That brings us to Chapter 6, discussing biochemistry of the skin, again, another chapter. But this time, we're going to talk about the anatomy, physiology, and biochemistry of the sudoriferous glands. And you'll see it's a lot of people are a little skeeved by sweat and sudoriferous glands, but it's actually an amazing and very interesting process that your body goes through. One of the major functions of sweat is to cool the body by a process known as evaporative cooling, and it allows us to do many physical activities that otherwise would not be possible in order to regulate our body temperature. So when we talk about sweat glands, we'll first discuss eccrine glands, because those are the most well studied and I would say most well understood. And then we'll bring the discussion to Apochrine glands. And eccrine glands are the glands that are all over the body, and they actually regulate body temperature. And you can see here on the right in this illustration, it shows you an eccrine gland and what it is. And it basically is composed of two principal components. One is that it's called the secretory coil, and that's this part down here. And then you have this, what's known as the duct. It's DUCT, and the duct actually goes directly up to the skin surface. And when you see beads of sweat, those are essentially, sweat that's coalescing from multiple eccrine gland exit points. The Apochrine gland, which we'll also discuss, is something different. And this is what's found in the axilli region, so in the underarm region and several other regions of the body that we'll discuss a little bit later on in this lecture. And these actually don't go directly to the skin surface. You can see they have a much larger secretory coil, and then the duct for these goes directly up to the hair follicle right above the sebaceous gland. So the Apochrine glands are the glands that, again, are in the axilli region, and these are responsible for the odorous smells that we associate with sweat. The eccrine glands, which are basically positioned all over the body, are not responsible for smells. It's basically a water-like substance that contains some proteins, amino acids, ions, and things like that. Also electrolytes. So as you know, when you sweat, you have like a skin surface that has higher salinity levels. But in general, the eccrine gland, when we talk about evaporative cooling, we're talking about the eccrine glands, and we're going to discuss, all these different things about both sweat glands and also the composition of sweat. So we'll talk about eccrine sweating first. And eccrine sweating is essentially, as we already mentioned, it's a physiological response to an increase in body temperature. And this is the principal way that humans regulate body temperature through this process that's known as evaporative heat loss. So if we're not able to sweat, and there are people that have conditions where their ability to sweat isn't on par with other humans, if eccrine sweating fails, you can actually undergo heat exhaustion or heat stroke or hyperthermia, and this could even lead to death. So it's pretty serious. It's a very important process. In general, humans have about 2 to 4 million eccrine glands distributed over their body. That's quite a bit. And just in general, if you look at this figure, the eccrine gland is about 30 to 40 micrograms. So it's relatively small, but the total body, when you add those all up, there's about 100 grams of eccrine glands in the body, and that's about the size of a kidney, actually. We can sweat about several liters of sweat per hour, and these can reach as high as 10 liters per day. And in general, the sweat glands are most numerous on the sole of the foot, and it's actually the least or the lowest density of sweat glands is found on the back, or you could say the dorsal region of the body. So let's get into the ultrafine components or morphology of the eccrine sweat gland. And essentially, as I already mentioned, you can break it down into two different parts. You have the secretory coil, and then you have the duct. And we're going to talk about three different cell types in the secretory coil. One is known as clear or secretory cells, and then we have also these dark mucoid type cells, and then we have myoepithelial cells. And the myoepithelial cells, the clear and the dark cells, the function isn't exactly known at this point. The clear cells are actually, when we say secretory, they secrete the sweat. And then myoepithelial cells are actually like sort of a closer to a muscle type cell. So I guess when you undergo like a contraction process or something like that. So this might be what's responsible for sort of pumping out the sweat in a sense. We also have the duct, which is composed of basal cells and luminal cells. And I'll show an illustration so that you can see what that really is. So the functions of the eccrine sweat gland itself, it essentially secretes an ultrafiltrate of a plasma-like precursor, and this is done by the secretory coil, and this is in response to acetylcholine. And acetylcholine, as you might remember from when you studied physiology or biochemistry in the past, it's essentially a biochemical in the body that is released by the sympathetic nerve endings, and it travels from the sympathetic nerve endings, in this case, to the secretory coil, and it actually goes there and activates the gland and tells it you have to produce sweat. So this sort of statement up here, secretion of an ultrafiltrate of a plasma-like precursor. Plasma, as you know, is referring to the blood. Ultrafiltrate just means a solution that's passed through a semi-permeable membrane with very small pores. So it means that it essentially contains only lower molecular weight type substances in it. What this is saying essentially is that somehow the blood is filtering through something in the secretory coil, as it's sort of all tied together there, and that filtrate is somehow becoming the sweat that you sweat out. But it's I don't want to simplify this at all by saying that it's sweat is not simply an ultrafiltrate of plasma, but it has that general form. It has additional components as well. Another key function, so we have this secretion of this ultrafiltrate substance, and then the second function of the eccrine sweat gland is the reabsorption of sodium by the duct. Now, whenever you sweat, you'll probably notice that you have sort of a salty skin surface, and the reason that happens is that as you sweat, it has to be, just for the whole process of osmosis, sweating is coupled with the transfer of these electrolytes or ions for this whole thing to occur. But in order for your body to function properly, you have to maintain your electrolyte balance. And the body does this by reabsorbing sodium and potassium ions in the duct region of the sweat gland. Now, the reason that you have a salty skin surface when you sweat is because the duct can't absorb all of the salt back, and some of it escapes to the surface of the skin. But this is an incredible process that occurs, and it essentially, as I mentioned, conserves the body's electrolytes. We're going to show some illustrations of the eccrine coil and the duct, and there'll be illustrations of the cells that are present. And so this is just showing you the eccrine duct again. And actually, so this is the coil region down here. And then as you get into the duct region, you can see most of the duct is this straight area, but there's also sections of the duct that are coiled that this is showing. But essentially, when you look at some of the cellular illustrations on the next slide, we'll show like just one side of inside the duct or the gland. So, you would actually have like the whole coating inside. So if you can just imagine, just looking at one face from within the duct. So just to clarify that a little bit, so we have these two illustrations here. The one in the bottom are cells that belong to the secretory coil. And again, this is like the empty space in the secretory coil. And then on this other side, way over here to the left, you would have like another cell that would be, or another group of cells facing over this way. So, if this isn't clear, all I can ask you is to probably contact me. But this should be pretty obvious to you when you look back at this slide, and then you look at this slide. And, and also the one for the duct, we're only showing one small section of it. So this is like the empty space of the duct in here. So it's like hollow. And then we would have, more cells on this side if we were to draw an illustration. And actually, this would be three-dimensional, so you would have, cells going all around here, and then, sort of like a cylinder of cells, and then you would have an empty space in the middle, and that empty space is known as the lumen, Lumen, it's shown here. We also have a lumen in the coiled region, so it's essentially the empty space that's there. So what kind of cells do we have in the secretory coil? If we look here, I mentioned before myoepithelial cells. These are myoepithelial cells that you can see here. They're attached to this basement membrane region that's shown here. Notice we have, just like we had in the epidermal junction, we have a basement membrane zone in this type of tissue as well. That's very common through many parts of the body for where you have epithelial tissue. But here we see that these myoepithelial cells, on their border, they have these sort of, this next cell here, you can see there's a C here and a C here. So this is one cell below, and this is another cell above, and these are both clear cells in this illustration. And again, the clear cells are responsible for making the sweat. In between the two clear cells, we have IC. This stands for intercaniculi, and I don't see it listed on the caption below, but it's inter, I-N-T-E-R, caniculi, C-A-N-A-L-U-C-C-I, and I'll mention what the role of this area is here in a moment. First, I just want to mention that we have the mucoid cells, the dark mucoid cells. You can see there's a D here. So here's one cell up here, another cell, a third cell, and a fourth cell. Again, these are the cells that border the lumen. So you would naturally think that these would be the cells that are responsible for essentially secreting the sweat. But that is in fact not correct. The clear cells are the ones that secrete the sweat. And you may ask, how do they do that if they're not in direct contact with the lumen area? And essentially it's because of this intercaniculi caniculi that are shown here, this is the pathway for the sweat to reach the lumen. And so the sweat is secreted from the clear cells to the intercaniculi, and then it basically leaves out through this little canal here and goes into the lumen. Another thing that you'll notice is that you have these little illustrations here that look like, I don't know, these elongated structure. These are supposed to be mitochondria, and it's showing that the clear cells have a lot of mitochondria in them, so they're very metabolically active, and that stands to reason because they're secreting so much sweat. You'll also notice that the back border here is drawn a little bit darker than the other borders within the cells, and that's to indicate that there's a lot of a particular enzyme present, and it's known as sodium potassium ATPase. And this is a very important molecule or enzyme that's essentially present in the cell membrane of the cells, and it's responsible for transporting ions back and forth across the membrane. As you can expect, as indicated by its name, it transports sodium and potassium ions across the membrane, and at the same time it does that, it breaks down ATP. So it utilizes ATP as an energy source, breaks it down into ADP plus an inorganic sulfate group. And again, remember I said that enzymes that end in ASE, they sort of represent enzymes that break things down. We also see in the duct area that this enzyme is present along the border of the basal cells, so in the membrane of the basal cells. And in the duct region, we have the luminal cells that are directly facing the duct. They sort of have these microvilli type structures here that's increasing the surface area of its contact there. And then, the sodium potassium ATPase, as you can imagine, would function to transport these ions, and this is part of the conservation process of the electrolyte balance in the body. So the secretory coil, we have the dark mucoid cells that border the lumen. We have the clear secretory cells that are located just above, usually they're above the myoepithelial cells, but they could also rest directly on the basement membrane. And when you have two or more of these clear cells that are touching, you have these intercellular caniculi. Check here for the spelling. I gave you the spelling earlier. I think I added an extra C in my spelling. This is actually correct on this page. And we see that the intercellular caniculi open directly up into the lumen. So again, the only parts of the clear cells that are in contact with the lumen are those parts that are connected to the intercellular caniculi. The myoepithelial cells are essentially spindle-shaped, and they contain myofilaments. So remember from, we didn't really study it in this class at any point, but when we talk about muscle tissue, you have thick and thin muscle tissue. Usually the thick is composed more of myosin-type fibers, and the thin of actin. So the eccrine duct, we have the first layer are the luminal cells. So these are the cells that border the lumen, and then the second layer are the basal cells, and then we have the third layer, which is actually a basement membrane. And the duct, as we already mentioned several times, is designed for efficient sodium absorption, and the luminal membrane has both sodium and chlorine channels. So if we talk about the clear cell, we already mentioned that it contains a lot of mitochondria. It also contains lipofuscin granules, and lipofuscin is a yellow-brown pigment granule, and it contains lipid-containing residues from metabolism in the cell, or it's actually digestion, and it's considered an aging pigment. So, usually if you find lipofuscin is found as it accumulates in the eye, and it accounts for macular degeneration, so damage to the retina actually. We see that in the clear cells that the cell membrane contains many villi. These are sort of those finger-like projection structures that you see. And this type of cell is responsible for secretion of water and electrolytes. So the dark mucoid cells have a smooth cell membrane. They have dark granules in them, but their function is actually unknown. The myoepithelial cells, these are contractile cells. They're smooth. They have smooth muscle-like characteristics. So when we think of smooth muscle, these are like involuntary type movements. This is non-striated muscle. Just some examples would be the GI tract, the respiratory tract, the erector pili of the skin, so the muscle that is connected to the base of the hair follicle. These are, again, involuntary type functions. You can't really control your sweating. It's sort of an automatic process by the body. So in the duct, we have the basal cells, which are very distinct from the luminal cells. They have a lot of mitochondria in them. Their cell membrane is rich in ATPase. And again, ATPase catalyzes the decomposition of ATP, which is adenosine triphosphate to adenosine diphosphate plus inorganic phosphorus. And we see that the cell membrane is involved in sodium pumping for essentially what we will refer to as ductal sodium absorption. The luminal cells have less mitochondria than the basal cells, so they also have less ATPase activity, and they have a dense layer of tonofilaments. So as you can imagine, that's sort of like a keratin type structural material, and this is next to the luminal membrane. So this provides sort of the architecture of the duct area, so that it's it doesn't like collapse in on itself. It actually has some structure there. A molecule that I'd like to mention is beta-defensin, and we find that in the lumen and the duct this molecule is present. And essentially the defensins are a family of peptides that are cysteine rich and actually low molecular weight peptides. And these peptides are antimicrobial in nature, and they provide the epithelial surfaces in general with resistance to microbial colonization. We mentioned sodium potassium ATPase already. This is an enzyme located in the cell or plasma membrane, and this pumps sodium and potassium ions across the membrane in opposite directions. And again, this process is coupled with the hydrolysis of ATP. ATP serves as an energy source for this process. So this just shows you an illustration of that. This sort of, I think this is orange or greenish color here. This is supposed to be the enzyme which is embedded into a cell membrane. You can see the phospholipid bilayers on both sides, and here it shows the conversion of ATP to ADP plus PI, and then you can see that sodium is going in one direction across the membrane, potassium is going in the opposite direction. So we already mentioned also that acetylcholine is a neurotransmitter that it's a neurotransmitter of both the central and peripheral nervous systems, and this is what elicits a sweat response in the body. When this signal is sent, it's sent to the sweat gland, the eccrine sweat gland, and it's also sent to Apochrine glands for certain things. There's also actually, in Apochrine sweating, there's other hormones involved as well. But, you can just see here a sort of summary of the sympathetic nervous system. You have the spinal column coming down here, so you have the primary nerve that's connected to all these things, pupil dilation, salivation, relaxation of the bronchi in the lungs, heartbeat acceleration. You can see stimulation of glucose production and release, adrenaline secretion, and also noradrenaline, inhibition of bladder contraction, and just general stimulation. And what you can see here is this small thing that's not really noted is the sweat behavior. This falls into the same category as these other processes. Again, part of the sympathetic nervous system. So eccrine sweat is composed of, it's a liquid-like consistency, and we all know that because we've all probably sweat at some point in our life, or at least hopefully we did. And it contains, in addition to sort of a water-like material, it contains inorganic ions, lactate, urea, ammonia and amino acids, proteins and proteases, and just some general other compounds. And these are histamine, prostaglandins, and creatine. Actually, creatine is important to give energy to muscles. It plays an important role there. The inorganic ions, this is sort of showing you sweat is formed in two steps. Secretion of a primary fluid containing containing isotonic concentrations of sodium chloride, and this is done by the secretory coil. And then we have reabsorption of sodium chloride from this sweat fluid by the duct region. Lactate, this most likely comes from the sweat gland area, and again, we indicate this because some of these compounds, like urea, for example, comes from the blood serum. The ammonia and amino acids, essentially we see that the concentration of ammonia is about 20 times higher in sweat than it is in plasma. So that means that this is probably produced in the sweat gland somehow. And we also find free amino acids that are present in the sweat. And then in terms of the proteins, most of the proteins that we see are low molecular weight proteins. The epidermal growth factor is present, mostly in the duct region. And you also find the cystine proteases and inhibitors of cystine proteases that are present in the sweat, and also you'll find it in the machinery for the sweat. So here are some structures to be familiar with. You're already familiar with, I think we had seen sodium lactate at some point, so it's just shown as lactate here. It very easily could exist in the sodium form because of the sodium that's present in this region. We also have urea, which is characterized as having these two amino groups, and then you have this ketone functionality here. Then we have creatine, which we already mentioned is important for muscle supply of energy. We have amine groups here, and you also have this amine functionality in the chain, and then this carboxylic acid portion here at the end. Histamine, which you're also already familiar with. Again, we sort of think of the immune system response in histamine, but we also find it in the sweat secretions. And then last, there's a group of compounds known as prostaglandins, which are actually secreted by the prostate gland. This particular molecule that's shown here is prostaglandin E1. You can see that it essentially has this acid functionality here, and then it has the two long chains that are connected to this 5-membered ring, which contains a carbonyl group and a hydroxyl group. So now that brings us to the Apochrine sweat glands, and the Apochrine sweat glands, this is a nice illustration to give you sort of a rough difference between the distribution of Eccrine glands and Apochrine glands. Now, please keep in mind that Eccrine glands are distributed all over the body, and even though it's just showing certain regions here, so it's showing the forehead, axillae, palms, and soles. They're essentially distributed all over the body, but these are some of the regions where they're located at that you sweat the heaviest. You have the highest production of sweat in these regions, and you can imagine when you become nervous, where do you become sweaty at? Under the arms, in the axillae region, on the palms, and also in the soles of the feet, and on the forehead, of course. And these are thought to be sort of natural defense mechanisms for, going back from an evolutionary standpoint, and to provide surfaces with lower friction, that maybe you could run faster or do something faster without really having friction on your hands. The actual exact answer isn't known why, but that's what's generally thought in the scientific community at this point. But again, this illustration is meant to illustrate the difference between Apochrine and Eccrine. And again, Eccrine is all over the body. Apochrine, though, is confined to certain regions, and mainly to the axillae, the nipple region, the anogenital region, also known as the perineal region, and the ear canal. So this is what I really want to get across here, is where the Apochrine sweat gland apparati are located. So Apochrine sweat glands, again, primarily found in the axillae and perineum regions of the body. They become functional after puberty, and this is mostly due to hormonal changes. And in terms of their functions, it's believed that they serve a function as odoriferous roles, and this could be for as sexual attractants, for example. I know most of you probably won't agree with that. Most people are turned off by odor, and especially underarm odor these days. But think back, 4,000, 5,000 years. Maybe this was something that was, something that was like a signaling mechanism from one human to another. They also could serve as territorial markers, or they could be warning signals. And again, this is because, these glands are activated when you are nervous or you're under stress or duress, and they could serve as warning signals to the group that there's an invasion or something else is going on. Again, they increase frictional resistance, and they increase to some extent evaporative heat loss, and not nearly. They don't function as the Eccrine glands do, but they do have, they might play some role in that. So there's one molecule that I want to mention that's really important in axillae sweating, and this is 15-lipoxygenase 2, and it's found that there are high concentrations of 15-lipoxygenase 2 in the secretory cells of Apochrine glands. And the lipooxygenases are a family of iron-containing enzymes that catalyze the dioxygenation of polyunsaturated fatty acids in lipids that contain cis-cis-1,4 pentadiene structure. Okay, that's a pretty hefty description, so let's try to make that a little bit easier. And these lipooxygenases, essentially what it says is that they catalyze the formation of fatty acid hydroperoxides from fatty acids in the presence of oxygen. So polyunsaturated fatty acid, polyunsaturated means, if you remember from organic chemistry, unsaturated fatty acids are fatty acids that contain double bonds. Polyunsaturated means that they contain multiple double bonds in the structure. And those types of fatty acids are susceptible to a process that's known as lipid peroxidation. And that's what's happening here, is you have these fatty acids forming into these fatty acid hydroperoxide structures. These lipooxygenases are actually catalyzing this process. So what that means is that possibly, some of the odoriferous roles could be played by the activity of this lipooxygenase molecule. When you form, oxidized fatty acids, these may be more easily broken down by bacteria, which are on the surface of the skin, because they would feed off of that. So the composition of Apochrine sweat, it's a milky and viscous substance, and it's important to note that it is odorless. It has no odor when it is secreted. But just to put you in sort of, or to compare and contrast a little bit with Eccrine gland secretions, again, these are clear. The Eccrine gland secretions are clear, and their viscosity is like water. So definitely the Apochrine sweat is different than the Eccrine sweat. And as I mentioned, the odor is not present when these are secreted. What does cause the odor is bacterial action on the sweat, and it's believed that things like these fatty acids that are broken down into oxidized fatty acids are what the bacteria are feeding on, and ultimately forming these odor type molecules. And just in general, there isn't a lot known about the composition of Apochrine gland secretion. It's very difficult to isolate it and sort of to be able to differentiate between Eccrine gland secretion and Apochrine, even though we know that the one is milky and viscous, the other one is water-like and clear. On a molecular level, it's difficult to know for sure if something came from one sweat versus another. And to sort of confound the problem even more, as you recall, the sebaceous gland feeds off onto the hair fiber just below the Apochrine gland. So you have components of the sebaceous gland that can also contaminate the Apochrine gland secretions. There's another type of gland. So we have actually 3 types of sweat glands. I saved this one for last because this is actually pretty important. It's called an apocrine gland. You can see the spelling takes a little bit from Apochrine and a little bit from Eccrine glands in terms of the nomenclature. And this is because these types of glands are intermediate between Eccrine and Apochrine glands. And they're believed to evolve from Eccrine glands in the axilli region. So these are primarily found in the axilli region. And what's known about them is that their duct and secretory coil are fairly large, sort of similar in size to what you find in an Apochrine gland. But unlike the Apochrine gland, which opens up onto the follicle, the hair follicle, the Apochrine gland actually goes directly to the skin surface, just like an eccrine gland. And, just to give you an idea, at the peak of puberty, at 16 to 18 years of age, these glands represent as much as 45% of the total axillary glands. So it's a very important part of axilli sweating. There still isn't a whole lot that's understood about these glands. I think that people are still working on elucidating the structure and function of these, and hopefully in the years to come we'll know a little bit more about that. So that concludes this chapter, and just in terms of the reading material that goes along with this lecture, there are some sections in chapter 1 that you could read to cover this. So I hope you enjoyed exploring the world of sudoriferous behavior with me. I certainly enjoyed sharing it with you, and I will talk to you next time.